Good afternoon. I'm Hilary Michelson. I'm president of the Robert F. Furchgott Society. And on behalf of myself and my vice president, Dr. Danny Rosenbaum, I'd like to welcome everyone to this year's Furchgott Visiting Professor Lectureship. So many of you know the Furchgott Society was established in 2005 to honor the legacy of Dr. Robert F. Furchgott, who was awarded the Nobel Prize in Physiology or Medicine in 1998 for his discovery of nitric oxide as a signaling molecule in the nervous system. Last year, we celebrated the 20th anniversary of that historic achievement with the permanent installation of the Nobel Prize and proclamation in the lobby of the Basic Science Building. And for those of you who haven't seen it, I hope everyone has taken a look at it, but if you haven't, you should stop by and look. I think it's a beautiful tribute to Dr. Furchgott's wonderful achievement. This year, we continue to honor Dr. Furchgott's legacy with this annual Visiting Professorship Lecture. And for this 13th lecture, we're honored to have Dr. Edvard Moser, who won the Nobel Prize in Physiology and Medicine in 2014. So one of the missions of the Furchgott Society is to bring well-known, prominent international scientists to our campus to give lectures and have roundtable discussions with members of our community. But the Society is also committed to supporting the research mission of Downstate by, through a number of granting awards and mechanisms that we administer to and award to young, developing scientists here on the campus. And so, and along with that, we've also developed a partnership with the Arthur Ashe Institute for Urban Health as a way to, again, support the development of young scientists from the Brooklyn area. Of course, all of that, I wouldn't be the president of the society if I didn't, do our usual annual outreach. All of these award mechanisms are underwritten by the generous donations of our donors and our members. And for those of you who are in the audience and have not become members of the society, I encourage you to do so. You're here, you're enjoying the fruits of our, our society's mission, so why not become a member of the society? It's a wonderful group that really does support the research mission downstate. Before I move on to the final, to the more formal programming, there are a number of individuals I would like to thank who work behind the scenes to bring these events together. First of all, I'd like to thank Rose Jackman, the executive director of the society, and her assistant, Roseanne Perceau, and the members of the support staff who've done all of the behind-the-scenes work to make everything come together seamlessly. Without them, I can guarantee you these events would not come off quite so smoothly. I'd also like to thank, a very special thanks actually, to our graduate student, and I'm going to try and pronounce this correctly, Swetha Frapkar. I got something like this, and I practice. But as someone whose name is always mispronounced and misspelled throughout her entire life, I understand, I feel the pain. So I apologize. But anyway, Swetha has been so instrumental in, again, escorting Dr. Moser through the process and helping organize all of these events behind the scene. So I really want to give you a special thanks as well. So it is now my pleasure to introduce Dr. Chris Roman and Dr. Jeremy Copeland. Dr. Roman is the chair of the Basic Sciences Review Committee for the Furchgott Society, and Dr. Copeland is chair of the Clinical Sciences Review Committee, and they're going to announce the winners of the 2019 Robert F. Furchgott Scholar Awards. So, Dr. Roman?
Hi all, this is a great day. I'll be very brief. I think everyone can appreciate the importance of supporting the future. And one of the great aspects of the vision of the Furchgott Society was to have a way to acknowledge the next generation of new scientific talent that will make the next set of breakthrough discoveries, groundbreaking work, through a spirit of passion for science, intrepid, innovative thinking, and who themselves might go on to win a Nobel Prize of their own, just like Dr. Furchgott. It's an honor to serve on this committee. I served with Dr. Habitova, Dr. Alcaron, and Dr. Perkins. I can't find you. It's a pleasure and an honor to be able to spend time with colleagues who you have such tremendous respect for and review truly exceptional science from amazing young scientists who've submitted their work for evaluation by the committee. And I think the best times that we have are really when we struggle, because the work is always exceptional. The creativity, the hard work, and thoughtfulness and potential for the future is there in all the applications that we receive. So I think I'll just sort of cut to the chase and say that after much deliberation and thought, we're very happy to announce Dr. Salvador Dura Bernal as the awardee of the scholarship. I will say part of the reason that I was so persuaded is because I was the only non-neuroscientist on the panel. And so I sort of had a higher bar to raise my sense of wonder and enthusiasm. And the excitement and interest and how much you taught me about what you can do with modeling, computer modeling, something so sophisticated and complex as the brain, was really wondrous. And I know that your future trainees and students are really going to benefit from your clarity of thought, how well you articulate such complex ideas, and your really invaluable sense of wonder at the process. So with that, congratulations.
Hi, my name's Dr. Jeremy Copeland, and I've been chair of the Robert F. Furchgott Society Clinical Science for the Clinical Science Award for the residents and fellows. And as was mentioned, we have the privilege of seeing excellent science coming out in our young residents and fellows. It's certainly a very difficult task to do research during residency and fellowship while there's clinical responsibilities. Nevertheless, the caliber of the applications were really quite remarkable, and we were struck by the level of science that addressed the underserved community in which we live and work. So on that note, I would like to make this award to Rochelle Mendoza. Dr. Rochelle Mendoza, are you here? Thank you.
Thank you so much. I'd like to introduce our distinguished professor, Dr. Todd Sackner, who will do the introduction of our visiting lecturer today.
This lectureship honors the memory and accomplishments of Robert Furchgott, who received the Nobel Prize in 1998. And today's speaker is Edvard Moser, who received the Nobel Prize in 2014, together with May-Britt Moser and John O'Keefe, for the discovery of the navigational system in the brain. So to prepare for these introductory remarks, it was natural for me to look at the Nobel website to read Edvard's autobiographical statement. So Edvard studied psychology as an undergraduate at the University of Oslo, along with May-Britt. And together they received a doctorate in neurophysiology, studying synaptic long-term potentiation and memory. And they brought the Morris water maze to Per Andersen's lab in 1995. He then went on as a postdoc to the University of Edinburgh to study hippocampal memory with Richard Morris directly, and then on to the University College London to learn unit recordings of place cells in the hippocampus with John O'Keefe. He and May-Britt then moved in 1996 to the Norwegian University of Science and Technology in Trondheim, Norway, where he is professor of neuroscience and director of the Kavli Institute for Systems Neuroscience. So as I was reading his statement, there was a single line that I was struck by, which was he had a mentor who, so that Edvard could learn more about the interface of psychology and physiology, he gave to them a special issue of Scientific American, which was published in 1979, which was all about the brain. And that struck a chord for me because some mentor gave me exactly the same issue. And so I thought it would be a good idea to just check out what's remained relevant for over these 40 years and what is not. So that's the issue. It has a nice looking brain. And so if you open up the issue, you see something that's pretty, looks, you can see where it went to. It looked, pretty good. And then they even had sort of black slabs that you could hold in your hand, and that will take you to the stars. And some things look good after 40 years, and some things look bad at 40 years ago. Okay. But the important thing, of course, is the table of contents. And so if we look at the table of contents, what we see is that the majority of the studies, and these are to the 20th century foundational studies, are really about the neuron and within the cell. And the attempts of like Hubel and Wiesel to look at how the brain mediates psychology and cognitive processes, they really hugged close to the shore, meaning they kept close to the sensory system or to the motor system. It was really only Francis Crick in the last article really speculated how the future of how the brain might understand cognitive and psychological processes. So there was really little cognition and little psychology. And that is where the breakthrough of Dr. Moser's contribution lies. Because while John O'Keefe discovered place cells in 1971, and Jim Ronk here at Downstate found head direction cells in 85, it was the Mosers with the discovery of grid cells, and then grid cells with directional tuning, and then border cells, and then speed cells. It was really Dr. Moser who, as my friend Andre Fenton says, provided the parts list for the first cognitive and psychological capacity of the brain to be understood, the navigation system. And thus, in this sense, his was the first true 21st century Nobel Prize in neuroscience. So, Dr. Moser.
Thank you, Todd, for that very generous introduction. Also, thank you to the Furchgott Society for choosing me to give this lecture. So I think this lecture is a nice opportunity to celebrate both Dr. Furchgott and the achievements that have been made here at SUNY Downstate, since his work was essentially all performed here. So I want to focus on a different part of science, and as you heard, this will mostly be about the neural basis of our understanding of space or our own position in the environment. So as Todd pointed out, our sense of space is different from what had been studied most of the time at the time when I was a student myself, which was essentially focused on the simpler early sensory and early and late or peripheral motor systems. But the spatial mapping system depends on parts of cortex that are very far away from both sensory cortices and motor cortices, and in the sense is in the middle of the brain. And for that reason, for a psychologist, it is a dream area to study. So I came into this area around 1990. But at that time, it was actually quite an exciting time because things were very, very different from the way it is now. Essentially, there were 2 or 3 labs who did all the work, and one of them was the Brooklyn lab here with Jim Rank, and also with John Kuby and Bob Muller and Steve Fox, and people who established this field to a large extent together with the O'Keefe lab in London, and provided the fundament that made it possible for younger generations, including myself or André and many others, to build on this and develop it into one of the most active areas of research in high-level brain functions. So I've been here once before to lecture about the navigation system. That time you had to wait. It was a different audience, but you had to sit and wait for almost an hour because I couldn't find a way to lecture. So this time I appreciate that the staff has provided me with cabs and guides to follow me from door to door so that I couldn't make no mistake. And here I am, so I'm glad that you didn't have to wait this time. But what I want to do now is to step back a little bit in time just to review the field very, very quickly up till today in the first part for those who, if anyone, doesn't know much about place cells or head direction cells or grid cells, and then I will spend the rest on a couple of quite recent observations and end with some very preliminary stuff that's still ongoing. But one of the real milestones in this field is, of course, the discovery of place cells. So place cells are cells that are active only at certain positions, and I'll just illustrate that by playing a movie, which I always do at the beginning of this lecture, just to get everyone on board. So what you will see here is a rat that's seen from above, walking around in a square box that is 1 by 1 meter, and there are chocolate crumbles thrown out into that box to keep the rat walking around for continuously for 10 minutes in this case. A cable is connected to the rat's head so that electrical activity from the electrodes or tetrodes in the hippocampus of this rat are then transmitted to a computer. So we will be listening to the spikes of a single cell here. And you can, these are the action potentials of the cell, and you can hear that this cell, and also see that this cell is active specifically when this rat is up in the left corner for this particular cell. So this is a typical place cell in the CO1 of the hippocampus. Can also be illustrated with a color code like this. So warm colors, red is high activity, and blue colors is low activity. This shows the average of activity over a period of 10 minutes. And you can see that this cell is active mostly, or actually exclusively, in the upper left part of the box. And different cells, different place cells, are active in different parts of the environment, and together, as suggested by John O'Keefe and Lynn Nadell in 1978 in their famous book, then together place cells were proposed to form a part of an internal map of a space, or a kind of map that was suggested already by Tolman several decades before. So, of course, the discovery of place cells led to a lot of research, but and especially because this is in the middle of the brain, so an obvious task was to find out what are the inputs that could probably give rise to such specific firing. But nonetheless, this was hard to understand until a second component was discovered. So what you see here is Jim Rank together with Jeff Taube, and I was told this is about 1987. So this is a 3 years after Jim reported the discovery of head direction cells at the Society for Neuroscience meeting. So a head direction cells is the second type of cell, functional cell that was found in the hippocampal, parahippocampal system. So Jim found these cells in what they call the post-subiculum, or which is also known as the dorsal part of the pre-subiculum. And these are cells that fire specifically when the rat's head is pointing in a certain direction. So on the x-axis here, you see for one cell the firing rate as a function of head direction, and you can see how highly specific the firing of this cell is. Unlike for the place cells, these different head direction cells fire for different orientations, so that also here you have the whole range of orientations represented, and you can, from the combined activity of many cells, infer easily the head direction of the rat. So then, by the end of the 1980s, two elements were known. There were the place cells, there were the head direction cells, and obviously this triggered ideas about how these interact. How do they contribute to the larger map? And that then gave rise also to a third very important development in my mind, namely the development of computational models. So then remember that this was one of the first brain areas that are not sensory, not motor, where you actually had some elements that you could put together and you can start speculating about how they arise and how they interact. So models for the interaction were developed in the, particularly the 90s, 80s, 90s, and this was then when May-Britt and I came into the scene, and our wish was to try to understand how could place cells arise, and also how do the different types of cells in the system interact. So what we did was to go into the entorhinal cortex. So this is the entorhinal cortex of a rat brain. You see a rat brain here and see it from behind. And the colored area is the entorhinal cortex. And the upper part here is the medial entorhinal cortex. So we started recording there, both because it is the major interface between hippocampus and rest of cortex, but also is a part of the same system that contains the head direction cells, although the head direction cells are in a neighboring region. So some studies, including work that was done here by the Rank Muller Kubi team, had been recorded in this area, but because we had teamed up with neuroanatomist Menno Witter, we then decided to go a little bit more dorsal in this area than anyone had done before. And that then gave us the opportunity to see some cells that hadn't been seen before, and that is the grid cells. So what you see here is, again, the box. You remember the rat that was walking in the box in the movie. The gray trace here is the rat's path in that box, and each black dot is the location of action potential, also spikes of that cell. And what you can see now is that, again, like for the place cells, there are regional or localized activity, but there are many clusters of activity, not one or a few, but many clusters that form a very regular pattern that repeats itself all over the space. And that pattern you can see here, where I have, in this plot, I put these red lines on top just to illustrate the nature of the pattern, and you can see that it is triangular, or actually, to be precise, a regular hexagonal pattern that repeats itself all over the available space to the rat. So it's like a grid that covers the space, and for that reason we then called these cells grid cells. So there were many grid cells that have slightly different properties. So this shows 3 typical grid cells recorded at the same time on the same tetrode. So this shows the electrodes, and then at that time, and still mostly today, recordings are done only at electrical signals are picked up only at the end of the electrodes because they are uninsulated. Here are the cells, and you see one red, one blue, and one green. And here you see the grid patterns of 3 cells that are recorded on the same electrodes. and you can see that they have what we call a different phase. That means that their XY locations of the fields are shifted quite randomly. So this is similar to what is sometimes called a salt and pepper organization in the visual system, so quite random distribution, at least in rodents, there as well, for a number of features, including orientation, selectivity, odors, and place cells. So there is some organization perhaps present at a finer scale, but largely this is still the case. That's very different when it comes to the scale of the grid cells, because the scale is organized in a dorsal to ventral fashion. So what you see here is a sagittal section through a rat brain. This is hippocampus, this is medial entorhinal cortex in color. And at the top here you have cells that have a small scale, so the grid fields are small and they're close together. And then the scale increases as you go down the medial entorhinal cortex to the end. And at the end, well, we have actually only recorded about halfway down here, but very clearly the scale increases, and at the bottom here you can have more than a meter between each of the fields, which makes it then impractical to record itself. This shows the spacing that increases with distance from dorsal to ventral as averaged across animals. However, if you look inside one animal, you will see that this change from dorsal to ventral is actually not a gradual process, but it is stepwise. So each dot here shows one cell. On the x-axis, you have dorsal to ventral. Y-axis is the scale of the grid. And you can see that when you go from dorsal to ventral, the scale increases on average. You can see that it is actually discrete values. So it's one here, we call module 1, module 2, module 3, and module 4. And this increase actually is in a quite specific manner. I don't have time to go into that. All right. So now a few words also as a background about how could such localized activity be generated. And obviously this is more theoretical and more in the speculative domain, but I still think it's important to include because it serves as some background for the later, the studies that I'll present somewhat later in this talk. So again, it began with head direction cells. And once the head direction cells were discovered, many groups proposed how they could be formed. And essentially, there's one common factor to all of them, namely that they are formed by what is often referred to as a ring attractor. So what does that mean? Well, if you imagine now, this is each cell here is shown as a circle in the brain of a rat, for example. It could be in the dorsal presubiculum or postsubiculum. They are organized in this drawing according to the direction of firing, so that two cells that are close have very similar directional tuning. Now, the idea is that there are strong excitatory connections between cells that have similar directional tuning, and then in addition you have weaker but more widespread inhibitory tuning to cells that are further away. The result of that is that you get localized firing in this network, because cells that have similar directional tuning excite each other, and then they inhibit those that are further away. And then, if you then add directional inputs or movement inputs, so in this case head movement inputs, angular head movement inputs to the system, then that bump of activity that's created by these excitatory connections can be driven around 360 degrees in accordance with the rat's actual direction of its head. That's essentially the idea behind models of head direction cells that were proposed in the 1990s. Now I'm mentioning this because this was the direct inspiration for models that were suggested for place cells in 2 dimensions just a few years later, and especially from McNaughton's group. So the idea now, you can see the similarity. So now each dot here, each circle is one cell. These cells are organized now according to their location in space. So this is not cells as seen on the top of a hippocampal patch, for example. It is cells that are organized according to where they fire in the box. So this is a box like the one that you saw in the movie. So, for example, a cell that fires in the upper left corner is here, and one that fires in the bottom right corner is here. So again, how to get localized activity like in a place cell? Well, the idea was that there are excitatory connections between place cells that have similar firing locations. And in addition, you have a ring of inhibition that is wider out. That sort of that prevents the bump of high activity to spread around and sort of get out of control and excite the whole tissue. So this is often referred to as a Mexican hat. So that's why I put that one up here, because you have the excitation locally and then inhibition in a wider area around. But again, just like for the head direction models, you can suggest then that there is input that refers to the animal's movement in space, like the rat moving here, that are the input about the direction and the speed of the animal as it wanders around in space, that then drives this bump of activity around according to the animal's location. So this was suggested for place cells, and this was mid-1990s, so there were only location-specific cells that were known at that time. But there were some challenges about the place, using this for place cells, and one of them is a phenomenon I'll get back to, namely what's called remapping, also discovered here, which is that you have very different place configurations for different environments. So to get this to work, in some sense, you would have to propose this for every single representation or for every single environment where place cells encoded space. So then came the discovery of grid cells, and it was then suggested that perhaps such a pattern could either also or in only rather exist in the grid cells. But then how to get such patches of bumps of activity to form a grid pattern or a hexagonal pattern? Well, you could then extend the two-dimensional model simply by putting them into a similar network. Still, the dots here, or the cells, are organized according to the preferred location of firing in space. But if you now have local excitatory activity and a ring of inhibition around, you can imagine, for example, that there are multiple such patches, and each of them have a ring of inhibition around. If there's such a ring of inhibition, they will inhibit each other, and you will get a hexagonal pattern just as a result of that ring of inhibition. And you don't even need the excitation. You can actually even have just the inhibition. So then it's not a Mexican hat any longer, but an inverted Lincoln hat. So this is not the only way you can generate it. But let me then just leave it here, say this is one way that you can generate localized activity in the brain, but it's essentially the same model in different ways proposed for head direction cells, for place cells, and then transferred to grid cells, which proposes that localized activity is generated by mutual excitation, and that this activity is moved around based on inputs about your own movement in space. So there have been other totally different other ways proposed as well, other models, but they have suffered more serious challenges. So this is still the dominant model, although there are many things that are not explained and is still has to be subject to research. But I provide this now as a background because I will come back to it. Let me just say, without going into it, that there are other types of cells too. So Todd mentioned some of them. Again, first, the head direction cells exist also in entorhinal cortex. I won't say more about that now. There are speed cells, cells that you can see here this is a trace of activity for 7 different cells, each in a color, and in gray in background you have the speed of the animal, and you can see that these cells change activity in accordance with speed. And there are also cells that fire specifically along the local borders of the environment, so we're calling them border cells. So one, for example, here firing only on the east border in this environment, on the west border in a different environment. So now I will switch to more recent data. So these data were published yesterday, but in case you haven't read it already, then I will spend some time on that before I move to 2 other recent findings. So we thought that, I mean, as André had mentioned, there are all these cell types, but at some point you may wonder, there can't be anything more. But the case is that there's one thing we hadn't considered, and that's, which is obvious, that the data that I showed you so far, they are all from very, very simple environments, and made so simple that we've taken out every single object in the environment. The rats or mice walk just on open surfaces. There's nothing in the box. And perhaps that also prevented us from seeing other types of coding that actually use these objects to encode space. And there are reasons for believing that animals actually use the relationship between objects to figure out their own location. And there's especially one study by Collett et al. From 1986 that tells quite strongly how important local objects in the environment are. So what you see here is again a cylinder. You see 2 objects here, the 2 circles here, and then a cross indicates a location where gerbils that walk around in this environment can find food that's hidden under the sand or bedding or whatever it was. So they trained on this task and to find the food here. But then, a given day, the 2 objects are displaced, pulled away from each other. And what the authors found was that now the animals searched at 2 locations defined by the individual reference or vector relationship to each of those objects. Either here, which is defined in sort of southeast, a certain distance from the first object, or at that other location defined on the southwest side, a certain distance from the other object. So that suggested that animals may actually use even individual objects and the relationship, distance and direction from those, to define their own location in the environment. This then gave rise to several ideas and hypotheses or models of how such coding could occur. I don't have time to go into that, but suffice it to say that we started recording in such environments with objects present. So what you see here is a mouse in a cylinder, and here's a Lego object put into the environment. So it's a tower, and here you see what happens. So 2 different cells, one in the middle row and one in the lower row. First, no object, empty box, and in these 2 cells, very little activity, just scatter activity. Then we introduce the object here and you see the white circle is the object, and here, and this shows what the cell behaved like. So this red activity shows that the cell now develops a very clear and distinct field on the north side of the object, some 20-30 centimeters away. If you now move the object in the 3rd column here to a new location, the cell still remains active on the north side at the same distance away. Same here, another cell that fires on the north side still fires on the north side of the object when you move it. And it's not, of course, not only applies to objects that that are on the north side. So this shows the distribution of orientations relative to objects for such cells. This shows cells, and yellow is high activity. So you can see that essentially every orientation relative to an object in the environment is present in the population. So these cells were all recorded in the medial entorhinal cortex, where you also have grid cells, you have head direction cells, or you have border cells, but some of them are then specifically cells that have a field defined by a distance and direction from the objects. And we call those object vector cells. And I forgot to say, Øyvind Højdal is the Ph.D. student who did most of that work. And distances, distribution within a box that can be up to 80 to 100 centimeters wide, up to 45 centimeters. So you have the whole range of distances as well. The identity of the object doesn't really matter. So you can see your different prisms here and different cylinders put together into the same box here. And you can see that the cells respond quite similarly to all of them. So look, for example, at cell number 8, the second one here. This cell fires on the southeast side of these 3 objects, and pretty much in the same way to all of the 3 objects, and the same for other cells too. Then we wondered, is there anything particular about the shape or the size of the object? So here, for cylinders, we increased from 2 centimeters all the way up until 30 centimeters diameter. You can see that here, and you can see that the cell still keeps its firing on the north side of that object, and also when they are present at the same time in the box. And same if you start out with almost punctured object and then extend it in 1 dimension to a very long wall, 62.5 centimeters in the longest case. So you see examples down here. So this cell, again, one that fires on the north side, the same cell as on the previous slide. And then you extend it, and the cell still fires on the north side, but quite often, typically then splits into subfields when you make the wall really long. And of course, this raises the question for those who are familiar with so-called boundary vector cells reported in, not in entorhinal cortex, but in subiculum, whether these cells actually are part of a same class. So one question that these studies raise then is, what is it actually that causes the firing? I mean, there's such a wide variety of objects of all kinds of shapes and sizes. So there are 2 possible classes of stimuli that could elicit the firing. So one is the possibility that is actually the fact that the object blocks the animal's path in the environment. It's something that prevents the animal to pass it. It's an obstruction to the path of the animal. Another one is that it is actually sensory features. So try to distinguish those. What we did was to lift the object so that they were hanging in the air, always 15 centimeters above the mouse, so that it could never reach up to the object. In that sense, the object did not block the trajectory of the animal, but it still had all its sensory properties. And for all of the cells, you get this, what you see down here, namely that when the object is standing, you have an object vector field, and that object vector field is maintained when the object is hanging on the wall. So it must be, for these cells, the sensory properties that elicit the firing. And that's quite different than from the border cells, because border cells seem to respond to the blockade of trajectory. So the data for that is that this is a border cell that fires mostly on the east side. Then we introduced a wall here, but we also lifted the wall so that the mouse actually could walk underneath it and go to the peripheral wall here. And if it's a lifted wall, it doesn't elicit firing on the left side here at all. But if you then put the wall all the way down so that it touches the wall and does block the path of the animal, then you get a field along that wall too. So it seems to be 2 different classes of cells. One, the object vector cells that respond to sensory properties of a wide variety of discrete stimuli, and the border cells that respond actually more to the blockade of the trajectory of the rat. Okay, so now let me switch to another property. So now I mentioned now properties of cells in the entorhinal cortex. There are many, many different properties of space that are encoded, but I still think that that's just one aspect of what characterizes entorhinal cortex compared to hippocampus. And there's one big difference between those two structures, and that is to the extent to which they exhibit what we call remapping. So remapping was discovered again here by, this shows a picture by John Kuby and Bob Muller, who did recordings in similar environments, circular environments or square environments. And here the color code is different. So the dark colors here mean high activity and yellow means low activity. So this shows an example from a cell. This is, I guess it's the same cell, right? That shows in one environment where it's a white environment, then the firing is on the left side and the right side here, and in the other environment, then it shows as a completely different field. And of course, in addition, so this is just an example of the fact that cells in two different environments, even often in the same location, can show totally different firing even at a different location. And this is even more interesting when you look at the population of cells, because if you compare, let's say you have 10 simultaneously recorded cells, then very often there's no similarity in which cells are active in one environment, which are active in the other. And for those that are active in both, there's no predictability from where they fire in one environment to where they fire in the other. So this tells something about the hippocampus, because it tells that in the hippocampus there's not one place code and not one spatial map. There are many, many of them probably. So this is illustrated here for two environments, but then we extended this to just demonstrate that if you now record in environments where you transfer a rat between 11 different environments over the course of 2 days, then you find, and you repeat some of the environments just to check for stability, then what you find is that the lack of similarity applies to all possible pairs of recording environments. So this matrix here shows the rooms that where the animals were recorded. So they're called familiar room 1, novel room 1, 2, 3, 4, 5, and novel 1 again, and so on until you get to the 10th novel room here. And then the same on the x-axis. And if you now look in the matrix, of course, along the diagonal, you get perfect correlation because it's the same environment. But what you should notice is that correlation is what is color-coded. What you should notice is that it is essentially dominated by blue. That means lack of correlation. So any combination of rooms here, if you take all of the cells and correlate their combined activity using population vectors, you find that, for example, between room N1 and N2 and N3 and N4, there is no similarity in which cells fire and where they fire. So this is just shows that the data that came from the remapping studies applies very generally and suggests that there are a very, very high multitude or very high number of representations in the hippocampus. So then, what about the entorhinal cortex? Entorhinal cortex is quite different because this is shown here for 3 grid cells. This is just a cartoon, but to make the idea, the concept, you see 3 grid cells with different phases here, one in green, blue, and red. Now, if you go to a different room, record the same cells again, you will see that the spatial relationship of the 3 is completely maintained as you go to the other room, so that the red one is just to the right of the blue one, just like it was in the other room, and so on. And this is shown for real data here. So again, population vectors, that means that you stack the rate maps on top of each other and then correlate each little spatial bin across the 2 environments. So in this case, we correlate 2 recordings in the same environment, A and A prime, or we correlate one environment with the other environment. And you see that you do get a grid pattern in these cross-correlation maps, which then means that the map is preserved, but you also see that it is shifted. That means that the XY coordinates may be different, but it still remains totally coherent, suggesting that the same map is used in the 2 environments. And the same applies to the object vector cells. So, for example, if you record a pair of object vector cells, here you see that these have about a 90-degree offset in their orientation to the object. So this is on the southeast side, and this is on the northeast side. Then it rotates as it goes to the other room, but you can see that it still maintains the 90-degree relationship, and simultaneously recorded head direction cells show the same reversal, which then matches early data from the head direction studies, which also showed that, that the relationship between pairs of cells is maintained. So I think what this all comes down to, when you compare recordings from different environments, is that the intrinsic relationships of the grid cells, of object vector cells, of head direction cells, they are all maintained. And for grid cells, at least so long as you record from one module of grid cells. So that may be different across modules, but I won't go into that. But I wanted to show this as an introduction to the second last point I want to show in this lecture, namely studies where we try to ask, now this similarity between environments, could it be because of similarities in the recording conditions on the environment? So one way to try out this, to test this, is to go to the extreme condition where you completely strip off the sensory inputs. And this is a study where we recorded from grid cells and head direction cells during sleep, because during sleep there is, if not absence, at least a very dramatic change in the sensory inputs to the brain. So what Richard Gardner did in our lab is then to record during slow wave sleep and REM sleep and see if the structure of the grid cells is preserved. And this shows data from an example data from a pair of grid cells that he recorded. So first, to the left here, you see two grid cells with an identical spatial phase. So it's one in red and one in black, and you can see that the grid fields are completely overlapping, and the same with the directional tuning. Now, what do the spike cross-correlograms look like? So this first, to the left here, you see a spike cross-correlogram from the running or the active state in the box. So no surprise that we see a peak around 0 seconds here, because this is, if the cells have firing fields exactly at the same places, you would expect that if cell 1 fires, then cell 2 will fire with an increased probability. But the more interesting observation is that if you now ask what is the firing relationship of those, this pair of cells when you go to the slow wave sleep state, you can see that you still maintain that peak. That means that if cell 1 fires, then cell 2 will also fire at an increased probability. And to a lesser extent, also in REM sleep, it's not the best example, but it is the same pattern. And conversely, if they're out of phase, that means the red one fires at different places than the black one, then of course you will see a dip in the cross-correlogram in the running state, but you also see that dip in the sleep states as well, meaning that those cells that fire together in the the active state also fire together in the sleep state, and those that do not fire together in the active state also do not fire together in the sleep state. And this can then be extended across the population. So these are all pairs of cells recorded, grid cells only. They are, what you see here is each line is the spike cross-correlogram. The color indicates the coupling between the cells, or a correlation if you like. It's actually the coupling using a GLM approach, which is slightly better than a cross-correlation, Pearson correlation, but anyway, shows the correlation between the cells. So for each line, you can see at the top here, so these cells are recorded in the running state or when they run in a box, and you can see that you have many cells here that are active or have a peak in the pair between the firing of the cells at 0 seconds, and then you have many cells that actually have oppositely correlated and have the dip. And they are organized or ranked according to how much or the size of the correlation at the peak here. So now, if you now ask what do the same cells do in the sleep state, you keep the ranking as it was in the active state here. You see that you essentially maintain the pattern, which then means that, and also in REM sleep, although a bit weaker, that essentially means that in the sleep state, those cells that are active together when they're running are also active together when they sleep, and those that are not active together when they run are also not active together when they sleep. And the same applies for head direction cells if you rank them according to the directional tuning, and also conjunctive head direction grid cells. So the head direction cells replicates the finding by Peirace et al. From then the Busacki lab. But together, all these findings suggest that there is a single map for position and direction that actually, to a large extent, still is expressed in sleep. And just to reinforce that, if you now ask, how is this maintained over time? So this is 1 hour of recording on each axis, the x-axis and y-axis. So this is a total of 4 or 5 hours. And it's binned, and for each bin, we define a vector, a population vector of the coupling values for all the simultaneously recorded pairs of cells in one rat that had 8 simultaneously recorded grid cells, and then ask what's the correlation between those coupling values at different points of time. And if they were different at all different times, you would just get a line along the diagonal. But you can see that high values, the high values is red, occurs all over, whether it's close or far in time, which is very different from what you get in hippocampal data. This is not our own hippocampal data, it's from a paper from the Busacki lab, but essentially shows with the same analysis that is dominated by white or low values of correlation, which then shows that in the entorhinal cortex, patterns of activity recur at different times, whether close or far away in time, whereas in the hippocampus, it is dominated by unique patterns that only very rarely repeat themselves. So, which is consistent with the idea that hippocampus is totally different in the sense that it shows remapping and alternates between very different patterns, whereas the entorhinal cortex does not. And of course, this remapping is what you want in a structure that encodes memories and should allow for storage of a wide diversity of memories and not just a single state. So finally, before I close, I want to say a little bit about these networks, because these data here, I should say that before I leave it, these data here are consistent with, I don't say they prove, but they're consistent with ideas that, that the activity is strongly predominated by the wiring or the intrinsic architecture of the network, which is, as you would expect, if it, for example, was an attractor network where the connections between the cells is what defines which cells fire together and which cells do not fire together. But one of the limitations of these attractor networks, or any network models, is that they're quite static in time. They don't allow for changes in time. And this is what I want to address in the final portion, because what can we say about how activity in entorhinal systems or any cortical system evolves over time? I mean, how does, are there trajectories of activity that are more likely than others, for example? So this is the work of Flavio Donato and Soledad Cogno and Horst Obenhaus, but with Flavio taking the lead. So again, what they did was that they tried to isolate sensory inputs, minimize them. So this time the animals were not sleeping, but it was mice, and they were running on a ball in a setup that's quite similar to the ones that are used in virtual reality in experiments in many labs. But the rats were running on this ball spontaneously. There's no reward, so there are many breaks. Sometimes they run, sometimes they don't run, and it's totally dark. No inputs of any visual kind at all. And then, while they do this, then they record many cells at the same time. So in this study, we didn't use a electrodes or tetrodes, but instead used two-photon microscopy and using calcium imaging. So the way to approach the medial entorhinal cortex is to use a prism that is squeezed in between the cerebellum and the cerebrum, so that you don't damage neither the cerebrum or the entorhinal cortex nor the cerebellum, and can actually image the surface of the cerebellum just as if it had been on the top of the brain. And then this just shows, maybe a bit hard to see, but you see the cells blinking here. So you can see up to 200, 300, 400 cells at the same time while the mouse is running on the ball. And then, so these are calcium waves. It's not action potentials as such, but still can be used as an approximation for many purposes. So how is the activity when the mice run on this wheel? So this shows first an example of the raw data. So here are neurons, a raster plot showing 200 plus cells over a period of a little bit more than 1600 seconds. And you can see that looks pretty noisy, can't really see any pattern. But if you do some sorting, things change. So the sorting is the method that we have primarily used, but not the only one, is principal component analysis. So what they did was that, first of all, they chopped the recordings into time bins that are very small, so for example 125 milliseconds, and then kept the cells ordered like this, so cells N1 to cell N, and then here are the time bins. And then they calculated the principal components using the time bins as observations and the cells as variables. And then for each principal component, they then defined the loading, or how much the cell was loaded on or correlated with, if you like, the principal component. So these are the original numbers of cells in these assembly of neurons, but they can then be reordered according to how much they're loading on this principal component. So the first one is the first one here, but you can see that, for example, the second one is also the same here, but then the third one is down here, 0.85, and then they get new numbers, cell numbers, according to the weights they have on this principal component. And what you then get, if you sort them by how much they load on the principal component 1, or even 2 or 3, then you get this. So it's the same recordings, but now ordered according to how they load on the principal component. And now you can see that a pattern emerges. So it's a repeating patterns, or repeating trajectories that go over and over and over again over time in this network when the mice essentially just run with no purpose. So how to quantify this? Well, first of all, try to break it down and denoise a little bit. So define, first of all, time bins. So time bins where we use different time bins, could be anything from 1 second up to 40 seconds. And the other thing was to break down the principal components in groups. So it could again be anything, but this shows an example where the principal components, so the 300 plus cells, were divided in 10 groups, so a little bit more than 30 for each group, depending on how much they load on the principal component. And so then you get this color matrix, and you can then ask which bins are more active than you would expect by chance, by just shuffling, and those are color-coded here. But the more interesting part for this context is if you now select for each time bin the one principal component bin that is most active, so you do that for time t plus 1, t plus 2, and so on, and then ask, is there any relationship between which bins are active at any given time, then this is what you find. So this is what we call a transition matrix. So you have the principal component bins on the x-axis and on the y-axis, and then color shows the probability of switching from one bin at one time bin to another one. And what you can see is a clustering of yellow colors along the diagonal, which means that transitions are most likely between principal component bins that are close to each other. And the same is if you only show it for those that are statistically significant. A different way of showing just the same is here. So here you have, if you now use 10 bins for the principal components and show them in different colors from 1 to 10, and then define, you have arrows between those that show transitions, then you can see that there are actually more transitions between bins that are near each other than you would expect by chance. So this is what you would expect by chance. So you have all kinds of transitions, for example, from 1 to 5, 1 to 7, or anything. But here you have more likely 1 to 2, 1 to 3. You see they sort of cluster around the periphery. And this is more clearly seen if you only pick out those that were statistically significant. So you see that there is a transition to neighboring bins. So then to the end of the argument. So if there are such transitions, then let us define possible sequences in these graphs. So here we know there are transitions from 1 to 2, or from 2 to 3, or 3 to 4, and so on, and even one from 3 to 4, 5, 6, 7. What is the total space of possible transitions? So that the longest transition sequence could be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, or 10, 9, 8, and down to 1. But then you have 1, 2, and 4, and all possible transitions. So now what Soledad did was that she went into the data and looked for all these transitions, frame by frame, tried to find them. And what she found is shown here. So this is, again, a recording of more than 1,600 seconds. And what is shown in blue is these upward transitions. So they repeat over and over and over again. And this is the downward, so the opposite direction, repeat over and over and over and over again. So in other words, there are sequences of activity that are stereotypical, they are directional, and they repeat all the time. But the most striking thing is the length, because look here at the time scale. These are, although they are never identical, they're statistically very similar, and they last for up to 2 to 3 minutes. They can be shorter, so here are some of the shorter ones. So in this case, the length is some tens of seconds, but 30 to 40 seconds is usually the shortest. And there seems possibly, and this is still not finalized, we're still working on it, but seems to be a relationship to age. So this is a recording from a 14-day-old mouse. This is from 22 days, 29 days, and 33 days. And you can see that at 14 days, there's never really any pattern. Can't find anything. 22 days, it starts to emerge. 29 days, you have long sequences in the order of several minutes. And usually it stops at around 30 to 33 days when you get them down to about 20, 30, 40 seconds. And this shows the number of sequences, how they evolve with age. And this shows the duration of sequences, which stabilize around P30. So this is interesting because a lot happens to the network at those times. So for example, with maturation as measured with double cortin and other measures, maturation occurs in layer 2 of entorhinal cortex around P20, 22, and CA1 comes later, 25, and then the network is completed only when the deep layers mature around P30. That's also when you finally get totally adult-like grid cells. So there may be a relation between that. So with that, let's skip that. Finally, just as a last slide, just say that the presence of such sequences may suggest then that we need to try to understand these networks, not only in terms of their spatial structure, but we also need to take into account the sequences of activity and trajectories of activity in this space. And there seems to be a lot that perhaps may be given by the wiring. You may say perhaps this could be related to the instantaneous behavior. So I didn't show data on that because we're still working on it, but it seems like these trajectories are not related to the running of the animal on the wheel, so they can transcend running and non-running periods. They involve most of the cells, so by implication not only the grid cells, and they're distributed widely in the network. So I think this is more food for thought because we don't quite understand it yet, but certainly the activity as it goes around in the network is not random and probably to a large extent determined by intrinsic factors and not only by the sensory inputs that come into the system. So with that, I think I want just to wish you, or thank you for attending and sitting here for an hour, and then let me say then, just at the end, that there are many people involved in this, and I haven't had time to mention them. Some of them are mentioned here. I did mention the ones that were involved in the trajectories and in object vector cells and in the sleep studies, and May-Britt, and funding from many sources, but of course there are many more who were involved in the older work. So with that, then thank you a lot for the attention, and thanks for inviting me to giving the Furchgott lecture.